Good morning, everybody. Thank you for joining us today. We'll wait for a few more people to pop on here. But this is our Buckeye Neurotrauma lecture series. We started this as sort of an outward-facing seminar. In conjunction with our neurotrauma research in progress, we have a lot of people that want to see professors' talks here in our area of neuroscience and neurotrauma. So we have quarterly Buckeye Neurotrauma lectures that are open to anyone who wants to join. And that's what we have this morning. And I'm really delighted to introduce our speaker today. So our speaker today is Dr. Giles Plant. He's a professor in the Department of Neuroscience and a member of the CBI program, which is the chronic brain injury program. Giles came to us at OSU in 2022, so he's been here just a short time, but figured to get a nice opportunity for him to highlight some of his work he's been doing in stem cells and transplantation and within the context of spinal cord injury and neurotrauma. Giles started his career in London, England, I believe, and then did his PhD at the University of Western Australia in Perth. Then he did a postdoc in Miami project, so he came to the United States for a bit, then back to the University of Western Australia as a professor, associate professor, then decided to come back to the US at Stanford University from 2010 to 2021. He was the basic science director of the Stanford University Spinal Cord Injury and Repair Program. Giles has been very productive over his career with over 60 papers in stem cell related journals like Stem Cell, Cell Transplant, but also trauma, experimental neurology, Neurotrauma, journals also like GLIA. He's had previous funding from DoD, NIH, Wings of Life, many other foundation awards, and had a lot of funding when he was in Australia. Very dedicated to service, has done a lot of study sections, including one on bioengineering of neuroscience, which sounds really interesting. Giles, you have to tell me more about that study section. And then I've been an editor of several journals or section editor, including Frontiers of Neuroscience and then Cell Stem Cells. Giles has hit the ground running here and has been very busy setting up his lab and being part of the CBI program and committees and various leadership roles here. So we're excited to have him and really happy that he has a chance to present his research to our group and everyone else who's joined in. So thank you for joining, and Giles, take it away.
Thanks, John. What I thought I would do today is just kind of give you a little bit of background about how we kind of introduced the induced pluripotent stem cell work. It was something I wasn't doing for a long period of time. And then when I went to Stanford, there was a lot of interest in induced pluripotent stem cell at the time, which is probably around 2011 in particular. And so what I'll do today is I'll go through some history of spinal cord injury in particular, just what the injury entails and what happens to patients, and then talk about how the cells are derived that we use and why we want to use cell transplantation and biomaterials and things like that. And then I'll give two examples of what the lab has done. One is an acute transplantation paradigm where we've transplanted within 14 days after an injury, which is pretty common in a clinical sense. That's the window that most people want to treat patients and things, is in that window, but also chronic as well. I'll mention we've done an entire chronic study using some induced pluripotent stem cells. And then lastly, I'll talk about some new data that's we've just is currently just about to be resubmitted back to biomaterials for publication, is some biomaterial combination with induced pluripotent stem cells in an acute model, again, of spinal cord injury, but importantly looking at some very tailor-made biomaterials that are injectable, which is certainly something that's very useful. Just want to say straight up, I have no conflicts. Just to let everybody know quickly, this is something that required to state, and I have no conflicts at all. So I want to mention this for an audience that may not know this. There was this issue of spinal cord injury goes back a significant amount of time, and it was first described going back in Egyptian times. And it's fascinating, I found when I first heard of this story, that John Bredsted, who's somebody who analyzed a lot of these scriptures from Egyptian times, saw this description. And it just shows you that in terms of spinal cord injury, how the injury itself has been around obviously a significant amount of time, but people have known how to describe it, and the aspects of losing sensation both in arms and legs. And unfortunately, in today's terms, these are the things that are said to patients currently, is that there's really not a lot that can be done, and they lose sensation, whether they get an injury in the cervical level cord or thoracic or lumbar. So it's just from an interest point of view, I thought for the audience that this is the first description that we know of is from this time. And I just thought, from my English background, people might find this interesting. And there's also an example of an American injury that you might not know of. This is a scene from the Battle of Trafalgar, where Lord Nelson, who you see lying down on the ground there, who was a famous admiral in the British Navy, was shot. And originally, he was thought he was just shot and he got injured through the heart, but in fact he was spinal cord injured. And one of the comments were from his surgeon at the time, which was, he described, "I've lost all power of motion and feeling below my chest are gone." And then there's Mr. Beattie was the surgeon on board. "My lord, unhappily, our country can do nothing can be done." And unfortunately, that's the kind of premise that a lot of people are given. Until, I would say, in the last 10 years, I think a lot of people have got a lot more hope about trying to get recovery from spinal cord injuries, which previously we thought, nah, forget it, write them off, you can't do anything. Another example is Patton. Now, Patton was an interesting one. He had a car crash 2 weeks, I think it was either just after the war, and refused treatment. He died 2 weeks later. Due to a fact of systemic infections that he got, but he refused to be treated because he felt that nothing could be done. So why? Well, spinal cord injury, I just thought I'd describe some of the pathology aspects, and I won't go into too much detail about the individual aspects because I don't really study a lot of the primary events, which is the initial mechanical injury. You get a loss of tissue due to necrosis. There's a lot of hemorrhaging of the blood vessels in the spinal cord. The secondary events are that you have distinct deflammatory cascades where the cells come in, clear out the debris. There's demyelination where the axons themselves lose their myelin sheaths and lose their conduction. There's also a cyst cavity formation, which is pretty pertinent to what I'm going to describe, because this is the issue. You've got a big, huge cavity space in the spinal cord that can't transmit axons across it, a little bit like a bridge, and using cars from one bank to the other, you need a bridge. That's exact analogy, though. The axon has to have something to grow on. There's a lot of glial scarring around the edges of this cystic cavity, which historically people have suggested that prevents a lot of regrowth spontaneously. And that's another reason we target this particular injury with cell transplantation. This is a human injury example. This is from my time in Miami, which Jonathan's mentioned. This is material that we used to get donated to our pathology area. And this is a C6 injury. And you can see the progression of the hemorrhaging of within the spinal cord and how much of the damage is caused from a level at C5 within your neck all the way through to like C8. You see the extent of the injury. Now, pathological condition, this continues to get worse as we the further away from when the initial injury was, and in some conditions can be quite large cystic cavities are formed. So if you can find a way of preventing this ongoing loss, it would be also another target for cell transplantation, which I'll mention a little bit later. As I mentioned, these are the events that happen at the acute and chronically. And then this is what the pathology tissue looks like if you cut it at a more microscopic level. And you can see the vast cavities and cystic cavities within the spinal cord parenchyma that you can see there. What happens when you have a spinal cord injury? As you can see here, I've just mentioned the numbers of levels. You get cervical cord, part of the cord, you get thoracic, you get lumbar, and you get sacral areas. And here, if you have the injuries, you can see what effects it will have on the patient once these injuries occur. I particularly focus now on the cervical division of the spinal cord, involved with head and neck movement, breathing, shoulder movement, heart rate, for instance, finger movement, hand movements. In particular, because now this has become the most common injury in spinal cord patients that we see. And so a lot of the focus you'll see today is on that particular injury. So the models we're using is a cervical injury model. So I just wanted to mention. And we look particularly at hand movement and finger movement, the ability to be able to grasp objects and move across the ground using those forelimbs. In particular, I wanted to mention here, now, there is a big difference in terms of anatomical sense between rodents and humans. What is important, I wanted to point out, if you look on number 1 and number 2, number 1 is the corticospinal tract. That lives as its projection profile from the brain in the more lateral aspects of the spinal cord. In the rodent, it's more in the dorsal aspects. So there is a difference, significant difference in terms of its projection profile between those animals. The cerebrospinal tract is another important tract that's a little bit less known about what its role is in humans, but in rodents it's particularly relevant for balance, and we've used that model previously. But I just wanted to show you the complex nature of the projections of the spinal cord axons that are in the cord, and trying to mend all these once injured, you can see is a difficult job. So you're going to need something that can stimulate a lot of different tracts to grow that have different functions. Now, the strategies to repair it, they're numerous, but I've put them into 4 little brackets here. Neurosurgery techniques in terms of stabilization of the spinal column straight after injury is a common one. So protect the cord from further injury from the bone shards and things, clearing those things out. They're very important. Pharmacological treatment. Historically, there's a lot of methylprednisolone was used for a long time, which is a steroid to prevent inflammatory responses and things like that. There's other treatments now which are being pushed onto the treatment through clinical trials, inflammatory responses and things like that. Neurotransplantation is a big part of this, which I've been a part of for the last 20, 25 years in terms of trying to find an appropriate transplant that we can use in the spinal cord once it's injured. And that can be numerous. I'll go into some details about the different cells that you can use. And also now, the advancement of biomaterials is a key thing, which I'll mention last in the talk. This is a real key. You can now have biomaterials that are either solid, have different softness that can match the areas of interest that you've injured, whether it's the brain, the spinal cord, peripheral nerve. They also can have active substrates placed into these materials that allow axons to grow and glial cells to respond in an appropriate way. But this also can be injectable, which I'll mention as the last part of the talk. Is a fascinating advancement in these materials that can be injected as a liquid, then gel within the actual body. And I'll mention that with some of the cells we're doing. Well, what is the role of cell transplants? Well, they're numerous. Replace the lost cells, as I mentioned, due to the injury. They can remyelinate those axons that were denuded by the initial injury and the oligodendroglia that normally myelinate and wrap their myelin around them to increase conduction. They're lost, but you could use, as some of the clinical trials have done, use these myelin progenitors to remyelinate those axons. You can also promote axonal regeneration in these circuits, which is really what I do in terms of using cells that promote axonal regeneration. Promote tissue sparing to prevent the further loss of tissue, which is also a real important area of a target for we use. And then another one is promote endogenous host response. Now, That's a stem cell response that people have looked into because there are endogenous stem cells within the spinal cord. Well, unfortunately, they either don't survive for very long and are not kept alive, and for the reasons that I think are a little bit unknown still. And then lastly, I was talking about scarring and inhibitory aspects. This is to modify the environment, and people have used enzymes such as chondroitinase to cleave off these inhibitory molecules, and these are the targets. I'll be concentrating mostly on the regeneration aspects. Sorry. Well, what are transplants? And I thought I would start with the stem cell side we have used previously. I think it's on this. What are the different types? Well, they're characterized based on the donor who provides the cells. Now, autologous cell transplant is something that refers to stem cells that are collected from an individual and given back to the same person. This can be stem cell transplants. It can be also glial transplants like Schwann cells, which have been used in the clinical trials, or in olfactory glia that have been used in clinical trials, taken from a patient and grown up in the lab and then placed back in. Allogeneic stem cell transplants or allografts refer to stem cells taken from one person and given to another. These are similar to like matching organ transplants. They're matched from that side of it. We also have syngeneic transplants, which refer to cells that are taken from an identical twin of the recipient. That's less common, obviously. I mean, I know a few twins myself, but that could do this, but the chances of those occurring across the globe is very small. And then you've got tandem autologous transplants, which are also known as double autologous transplants. It requires the patient to undergo 2 stem cell transplantations within 6 months. Sometimes that's graft versus host disease, for instance, if anybody's heard of those things. So those are characterizations from that. I wanted to mention here is that these are talking about just cells generally. You can place them into the spinal cord in all sorts of different ways. One of the ways I did in my project, and it's been used clinically, is using tubes that you can place these. So they're mixed with the Schwann cells are mixed with Matrigel, which is not a clinically approved matrix, but what happens with that particular thing, if it's kept cold, it's liquid. If it's then put in at body temperature, it forms a gel, and you can put the cells in. These then bridge. If you can see on the right-hand side that I'm facing here, you can see that the tube, and inside that is a growth conduit where the Schwann cells have stimulated tissue regeneration. So there's examples of cells that can be used. This paper intrigued me back in 2012, and this is why I'll get into the induced pluripotent stem cell side of it. It was done by Mark Tazinski's group with Paul Lu in particular, and they looked at long-distance growth and connectivity of using neural stem cells. But they particularly also used a little bit, about a year later, used induced pluripotent stem cells generated from a person who was about 84 years of age. I think it was even in the title. And this was fascinating to me because they'd shown that they could engineer the cells to have GFP, so they could visualize them, and then transplant them into a complete transaction site or a contusion site, and then looked at this incredible extensive growth that these cells within the spinal cord. And that intrigued me from a point of view of, well, what could happen with our induced pluripotent stem cells? How can we use them? Is it safe to use a transplant that's undifferentiated? Do you need to take them into a neural stem cell period like they were doing? Or do you need to go a little bit further and push them into a type of neuron itself, which is potentially a little bit safer? The issue I found with a lot of these transplants was the number of factors that were combined with these cells. That, to me, you would never get that clinically approved. It's too many different cell types. And so for me, an interest from my lab was to reduce that probability of using all those different cell additions, but also give a singular cell type that you could use potential for allowing for repair and accord. Now, iPSCs can generate different cell types. It's been known for a long time, since they were first made by Yamanaka in 2006, that you can generate, for the nervous system, astrocytes, microglia, oligodendrocytes, motor neurons, cortical neurons, sensory neurons. These are successfully done by using cells from an adult patient or a young patient and you can derive these by reprogramming using 4 factors. You can then use those cells either in transplants, or you can use them in culture experiments to look at drug efficacy and things like that. So they're incredibly powerful from that point of view. The potential is large. Do you use the cells as an undifferentiated transplant? Do you use them as a neural precursor population? Or do you really, really want them as a differentiated cell? Now, you'll find from my next few slides, I've gone towards the more differentiated cell. So avoiding the complex nature of how we understand what happens with neural precursors. Now, a classic way of doing this is you take a patient's cells, you put 4 factors that Yamanaka found, which is c-Myc, OCT4, KLF4, and SOX2. You can put them into these cells, whether it's a skin biopsy, which is the most common, or blood cells. You can reprogram them into specific iPSCs from the patient. The key thing here is that you can use viral vectors to do it, or you can use mRNA. We've generally used mRNA to do this because it's safer and doesn't integrate that particular full gene within the genome. It's done episomally, and so therefore when the cells are triggered to become iPSCs, they no longer carry that information long term that originally triggered it. So that, to me, is a lot safer. You can do 2 different ways. You can look at therapeutically, you can differentiate them into an affected cell type, do screening compounds, disease-specific drugs, and things like that. We can also go to repair faults that maybe, say, the person has a certain disease aspect. You can repair that by using viral vectors or using aspects to repair that fault in that gene, and then put those healthy cells back and then hope that you can transplant them and reduce that issue. Now, we went from iPSCs, and we had an idea that we wanted to look at particularly a tract that's very important called the corticospinal tract in humans after injury of spinal cord. Now, we wanted to try and make these corticospinal tracts neurons from iPSCs, from human iPSCs. There was some information out there from particularly the Livesey group, you can see down here described, where they generated these cells according to particular identified genes and transcriptional factors that Jeff Macklis and Paolo Wallotta, particularly at Harvard, had generated. We have looked into this for a long time, since I was at Stanford in 2001 when we first started this. In 2011 when we first started this. It takes a lot of time to generate these cells, and I'll go into some of the description. In basic things at the bottom here is you have this SMAD inhibition initially, and then you go into you basically go into other aspects of the noggin cycle, and then you go into TGF-beta inhibition. I won't go into too much detail about that, but basically it's a quite complex addition of these factors that you add at particular times of development and differentiation through these periods from neural stem cells all the way to neurons themselves. But we thought this was a really solid idea to be able to do this, because the corticospinal tract does not regenerate currently, and we wanted a way of potentially using new ones. The big issue is that they're very diverse in terms of their population, and this has become a little bit clearer now with really lovely work done by Vibhu Sani, who was in Jeff Macklis's lab, who's now in New York, where they've begun to understand the corticospinal tract can be put into different subsets that can project to the lumbar cord, the cervical cord, and beginning to now understand that. Those genes are now matched with some of our human cells. And we're now beginning to understand and really understand what they do and where they project to, and see if we can get that information into the human as well. This is mostly done in mouse. So, well, what we wanted to do was to say, can we make them? Yes, we can. We developed an in vitro system over 2.5 years to develop this system. But then we wanted to look at regenerative capacity of these cells if they were transplanted in the spinal cord, and to learn more about how they connect. And then, do they have synaptic connectivity, and do they form graft activity that can therefore repair the cord after we've transplanted it? The theory behind it is that the connections to the corticospinal motor neurons are generally direct from the where they lie in layer 5 of the cortex, and they project down into the cord and synapse onto normal spinal motor neurons, and then control your gait or your movement in your hands and various other things. What we had, the idea was that if we can get the corticospinal neurons to be transplanted into the cord, it might act as a relay graft, and that neuron could act as a connection to the host corticospinal and then direct its growth to a motor neuron down distally from the injury site itself, and therefore form a new circuit. And that was the principal idea behind it. And the key thing was that corticospinal neurons grow for centimeters, not microns. And this is a key thing why we use this. Critical gap in a human is a centimeter. Using propriospinal neurons or other neurons, sometimes they will not grow that distance. And so we wanted something that could bridge that key gap, and this is what we thought would happen. I wanted to mention about the lesions. There's different subtypes of lesions and things like that. We used in this model, and I'll describe now, is a hemisection where we cut the right-hand side of the cord from the top to the bottom. So that right-hand side is completely deafferented. Other injuries are contusion injuries where you use a weight to drop down, or various other injuries like that. But ours initially, you'll see here, is mostly from data from a hemisection where the right-hand side of the cord is cut. So what we do is we have a healthy system. We then injure it at the cervical level, as I mentioned. We cut it from the dorsal to ventral aspects on the right-hand side of the cord. And then we place the transplants, which are the corticospinal neurons derived from iPSCs from human patients, whether male, female, and multitude. We have about 6 lines we've used. And can it act as a relay graft? This is just some of the front-end information that we use, and just to describe how we do it. It's complex, but the aspect of how the timeline is there on the top, where we go from human iPSCs to cortical neural progenitors, and they express factors like PAX6, SOX2, FESF2, and nestin and FOXG1, for instance. If you then let those mature through Wnt inhibition over the next few days, and then replate them onto surface of laminin and polylysine, they mature into these CTIP2, TBR1, NCAM positive cells, which are maturing dorsal deep cortical neurons, these layer 5 neurons. And we did some analysis. This is a paper that's submitted with looking at RNA analysis. But if you look on the left-hand side in C, you'll see the CTIP2 positive neurons, which are beautifully labeled there on the left-hand side, stained also with blue with TUJ, so they're neurons. We also get a electrical activity. I don't know if the video will work, but what we wanted to show was these are firing neurons that are from an line called 1815, differentiated into these cortical neurons using a GCAM6 fast vector that allows you to see these neurons firing over time. The interesting thing is, if you leave this a lot longer, it becomes more of a synchronous firing, which is particularly seen in the development of the corticospinal neurons when they develop. So it follows the same thing. So intrinsically, these cells are exactly the same. This is a quick draft of what we did. We used the C5-C6 unilateral hemisection. We did some tracing, which I'll explain, and some graft ablation, which I'll also explain a bit later. But we transplanted this at day 14 after the injury in a scaffold, which was fibronectin, set by thrombin being put in. But we also used growth factors or not, depending on the fact that Mark's group had used these factors. We used a subset of these and thought, is this really needed for these more mature neurons, or is it just needed for the neural stem cell side? So we did do that. These are some examples of some of the graphs, and I'll quickly go through this because I've been talking for a while. They're not consistent. Depends on obviously the injury itself, but what we do notice is that when you transplant these neurons, they will fill the cavity and stop. They don't migrate throughout the cord. They don't form tumors. They don't go to the brain, which the neurostem cells have been described to do. They're basically restricted by the size of that injury. And so you'll see different sizes here described in this particular panel. You'll see the graft is different sizes. It's because the lesion site's different. But what is interesting is they do completely fill the cavity. What we don't know is what proliferation occurs from the progenitors that are in these grafts. We're not fully mature only. There are about 80 to 85% of those cells we transplant are mature neurons, but there is a 15% progenitor population in there. So do they proliferate, or do they mature, fill the gap, then stop? And these are examples that we saw that you look, the bottom here, the counts of the cells are very restricted within the site itself in the center of the cavity. They do not migrate up and down the cord, which is, to me, is a positive. You do not want these cells migrating up and down the rostral and caudally to the cord. I think that's dangerous. It's also, I think, long-term going to cause issues, which has been seen in other grafts. Importantly, with this graft here, this particular picture here, if you look on the top left-hand side, if you look at the growth factor ones and the scaffold, you look at the volume, it's slightly bigger than when you don't use iPSCs or these deep cortical neurons derived from iPSCs with the scaffold. The graft is larger. That may be because there is some stimulation of proliferation due to these factors. What we found eventually was the growth factors aren't needed. They're just not needed. We found that the deep cortical neurons, if you put the iPSCs from these, if you put them in the cord, they do just as well without the factors, which is a difference to what neurostem cells are. They will not survive. So that was a real positive when we found that we can actually do this without any factor initiation. The counts of the cells, if you look at percentage aspects, they're all centralized, as I mentioned before, to the center of the lesion. Well, what we also saw was these are GFP labeled. They're engineered to have GFP, and they project axons mostly in a caudal direction, not rostral, which seems to be that they understand that they're supposed to grow down the cord, not up. And so that was a real key that intrinsically knew to do this. And this is 12 weeks post-transplantation. If you look at the axons here, the graft epicenter is shown here in C5, C6. You see axons here, 15 millimeters caudally. We also had the largest one, which is 26 millimeters, which we've seen growing. So these cells know how to grow and how to bridge a lesion. And do it very effectively and consistently. And that was, when we did transplants, we got 85% of the transplants survive. Comparison, if you look at the neural stem cell transplants that Dzinski group do, they're 25% effective. And that's something that's a vastly different result. And I think this is why we're so keen on using differentiated cells rather than the random stochastic aspect of using neural stem cells. The also important thing, we left a few animals over a year and we thought, well, are they going to survive? I hope so. And yes, they did. And to our delight, they were still present a year later, integrated within the nervous system, within the spinal cord, and expressed HCAM, which is NCAM, which is a human-specific NCAM, showing mature neurons within the cord that it had lasted all this time. And they're also expressed with SC121, which is a marker as a cytoplasm of human cells only. So we know that they survive for that period of time. These are some of the axons that you get. The red is a marker for SC121, showing the axons going down to 1.9 centimeters. And you can see these axons, once they go, they go. And they were able to project quite long distances down the cord. Other thing we looked at was where do they project to in the cord in relation to the existing tracks that I talked about earlier. And what we found was that the large number of these corticospinal neurons that were transplanted in the cord at C5 project in regions dorsally, where they normally project. If you look here, the corticospinal tract in a rat is normally here. In blue is where the projections are from our graphs. and they project in these large areas dorsally and also ventrally and laterally, where the axons were traveling. And as I mentioned before, you see large numbers of axons. Mostly the axons are within 6 millimeters, the largest number of these axons. You probably get maybe around 25 to 50 that can project long distances. So there's a difference. And this is why we said about Sarni's work and Jeff Macklis's work. It's important because they're beginning to understand that these axons have different transcription identities and project to different regions with these identities. And so this may be the case here, that these cells have different identities. Also wanted to say is that they express particular factors, and the good thing that we found was that these cells are stable for up to a year, but they do not undifferentiate. What I mean by that is they stay in their stable phenotype. They express the markers that are deep cortical neurons, identify as corticospinal, which is CTIP2. Stathmin was another one we found, which was quite unique. There's one description by one paper that described it as being like an immature marker for corticospinal neurons. Ours did express this. So whether they're fully mature is unsure at the moment. And that's something we're going to be looking into. But they express Stathmin and correct markers. SAPB2 is downregulated in our cells, which is correct. That's what they should be doing. The interesting thing is we have no longer any progenitors, no PAX6 SOX2 expression after 12 weeks, 24 weeks, or a year later. There's none. They're all mature markers that these cells express. In terms of, sorry, I've gone back again. I also wanted to show you here is that we did some analysis looking at our transplants, looking at the glial response, the microglial response. And what we did see is that the transplants over time, they had a less aggressive microglial response to these transplants. Why? I'm not sure. We know what the secretome is of these cells. I'm not going to show it here. It's something that we're still working on. But we know that there are particular secreted molecules that do interact with microglia and actually have positive and some negative effects on microglia, but this seems to be a very positive effect in this sense that it's dampened down this microglia response from staining only. That's all I can say, but it is an interesting thing we saw. This is what I was saying about CTRP2, which is, and TBR1. CTRP2 is in red here, and the pink is TBR1. And these are graphs 24 weeks later after we transplanted them, showing that the cells are still positive for those markers, and also GFP you see here. I've downplayed the GFP expression so you're able to see the other markers. This whole graph is GFP positive. Also, what these cells do is they stimulate long tract regeneration. They stimulate corticospinal tract regeneration, which is phenomenal to see. They also stimulate these cells to grow into the graft and out and innervate the lower motor neurons. In terms of its circuitry, that's something I didn't do in this particular experiment, but we're starting to look at this with some pseudorabies viruses to look at the whole synaptic contact of both rostral and caudal with these transplants. So that's something that we need to still do. Also, raphinergic responded massively to the raphinergic projection, hugely responsive to this. What we did do is use an antibody that Jerry Silver had used to put into the animal to block the serotonergic signaling, but this did not get rid of the behavior, the improvement in behavior, which I'll show you just after this. And that was an interesting finding, is that you got this large regeneration of serotonergic fibers, but it didn't affect the behavior. Whereas if we do, I'll explain a little bit later, when we deplete the neurons by using diphtheria toxin and take the whole graft out, all the behavior was lost and the improvements. I'll skip that one. This is just an example, and I'll concentrate on the top four graphs mostly, is that we use different behavioral measurements, particularly the pellet retrieval task. If you look here from 0 to 12 weeks, how significantly better the animals did, and we're able to retrieve a lot more pellets and significantly. And this is a particular behavioral measurement that is used by the animal lying flat with 2 staircases either side, and they're able to use their arms and their fingers to pick up pellets that are on the staircase either side. A normal animal can't do that after it's injured. These animals were able to do that significantly between 9 to 12 weeks after the injury, and the transplant. So this wasn't just straight away. This was something that developed over time, and the connectivity would be one of those things that we're interested in. Grip strength, I won't go into too much detail, but it was a rather bizarre result, whereas it seemed to be that the animals that were not treated couldn't let go of the bar. They almost had more spasticity. And so this was something I'd seen in Huntington's animals. When you look at it, and I talked to some of the researchers doing it, we found this with our spinal cord injured animals, that if they weren't treated, they couldn't let go of the bar. So it gives you an opposite kind of strength reading, which I thought was quite intriguing. Here's the diphtheria. We used diphtheria, full-length diphtheria toxin, to deplete the cells, which we thought was a great thing to do to try and understand how effective these grafts were. And if you do deplete them, the astrocytes are left there, the neurons are got rid of, they're taken out. But all of the behavior that we saw recovery-wise, which we did in 5 different measurements, all disappeared, suggesting that these cells have a significant role. We have moved these cells now into acute and chronic contusion. What I've been doing since I've been here, we've done a whole analysis now of chronic contusion. And what I can tell you is that there was 72 animals done, all behaviored, and the analysis now is showing some positive results, even with chronic transplants put in 3 months after the animals were first injured. And these are showing some of the transplants that we've seen in these chronic and contusion injured animals. And they also are highly vascularized, beautifully vessels growing in with these grafts. They're incredibly healthy. Part of that we think is related to the VEGF secretion by these neurons, which we've measured via proteomics. And we're looking into blocking that and seeing if that affects the ongoing success of these grafts. What I wanted to lastly just touch, I think I've got a couple of minutes, 5 minutes just to go over this. This is some new data that I've done in collaboration with Sarah Haleshorn at Stanford, which is new data that we've derived using these deep cortical neurons in combination with a very unique shield biomaterial. The use of this material is key because you've got contusion injuries is the most common in humans, but you've got to be able to inject this without and treat people without making further damage. So one of the options is that you can put the cells in an injection paradigm, but what with? She was developing a lot of these very unique materials that combined a C7 engineered protein with these peptide PEG copolymers. And the beauty of these is that the hydrogen bonds that form is that when you put them under pressure, they form liquids, and then you can inject them. But then when they're not under pressure, they gel. And so if you can mix the cells in with these particular gels, and also the fact that they provide a protection from the shearing effect of the injection itself, is a very, very cool thing to do. You can also change the modulus of these gels to match what the spinal cord is following injury, and be able to match those particular moduluses. Again, this is a repeat of what we did. We showed the whole aspect of how we developed the cells to put in. We also put some sequences, the RGD sequences and some scramble sequences that are known to help adhesion to these gels using the cells themselves. And we did a lot of different combinations and materials with modulus, and we found that 0% or very, very, very soft gel was the best one to use, and also the fact that RGD sequences was also significant in improving the cell survival and integration. We also want to look at some of importantly, the biodegradation of these particular gels. What does it do? Where does it go? And these are a lot of the biomaterials data that you see in CNS. Never describes any of this. And you want to know that it's actually safe. Where does it go to? And so we did a biodegradation study, and you can see on the bottom panel here that when you're looking at C7, which is, you can use an IVIS whole animal analysis, you have to open up the rat. This is the difference to the mouse. You have to open these the skin up and some of the lower structures because of the penetration of this particular type of fluorescence. The mouse is a lot smaller. You're able to do this, whereas the rat you can't. And so we opened the animals up to be able to visualize this. But what we saw that is the C7 only, they disappeared very rapidly, but when it's combined in a shield with the PEG, this signal continued on for a long period of time. So it was keeping the shield in the appropriate place and not degrading for a long period of time. Again, showing the shield again. But what we also wanted to do was to say, well, can these mixtures of cells, the deep cortical neurons with the hydrogel, be any different or better or not worse or what it was from the fibronectin? And what we saw was that the growth length was not as long, but we had good survivability with this particular shield design. And the cells again stayed within the lesion cavity, which is what you want to bridge that lesion. And there was shield was significantly better in terms of the graft volume was smaller than the saline ones, but they were concentrated more in that site of interest. The stem cells, on average, the numbers of cells were 5 times 10 to the 5, and on average, that's a significant amount of survivability, which is also good. But we saw key here was the Ki67 SC101 survival. The Ki67 measures the proliferation. That was significantly reduced in the shield. So it's safer, be able to have less progenitors proliferating within that. So that's something to consider for future stuff, is that you have materials that actually can reduce the proliferation and maybe on a time basis and make it safer for the person long term. The axons traveled for over 6 millimeters to 8 millimeters. It was reduced from what we saw in the fibronectin grafts. Why? Not sure. That's something that we need to look into and find out what occurs that. But it gave us an important aspect that shield can be designed to be put to a patient-specific design, whereas fibronectin is a general produced fibronectin from human sources. Safety-wise, could have issues. Matching the patient itself and being rejected is also an issue potentially. It's why a lot of things like Matridol cannot be used clinically. This is why it's exciting to be able to use a biomaterial that's matched the person. Behaviorally, we also saw improve, and this is only over 10 weeks. This is another thing. Previously, I'd used 12 and 24 and a year's worth of data. So it clearly shows that 10 weeks is a short period of time. And if you really want to see improvement, you've got to do these long studies. We saw improvement in grit strength, we saw improvement in right paw use, and we saw improvement in step errors in all of these when these cells were combined with this biomaterial. So that's exciting from that point of view. What is interesting is, and I'll wrap up now, is differentiation of these cells is key. We use a window that's from 35 to 50 days in culture before we use them. We've done a lot of work on knowing that this particular differentiation protocol is used that way. And these cells, if you take it out at 155 days, you will see glia start to appear. You will start to see upper level cortical neurons appear. So that tailored graft is key, that you use it in a specific time point after you do these differentiations. That's something I just wanted to mention, is that it took a lot of work to find that window, but once you found the window, you can really, really exploit it. Biomechanical clues, like I mentioned, is that with these biomaterials, with these cells, we used a very, very soft gel, not Shield 2.5. So, and it really is key that you have the right sequence in these gels to be able to get these cells to survive and integrate within the gel itself. And this is what the cells look like in vivo. And I mentioned about at the bottom left-hand side is you can see KR67 and SC101 showing that the shield reduces proliferation considerably. And these are examples on the right-hand side of the big, long grafts that these cells, when mixed with the shield, stay within the lesion site and successfully survive the transplantation, which is a key thing for us to do. So I there will wrap it up and just say a big, big thank you to particularly Vanessa and Jim, who were in my lab at Stanford, and Laura, who was in Sarah's lab, who did this biomaterial stuff. The lab now at OSU has processed numerous people. I can't thank but Ethan and Cole and Morgan and lots of different people have helped me process and work through the chronic data, which we're excited about finishing off this year and trying to get it submitted. So, and without the funding from the sources I've mentioned here in pink, it would never have happened. So I do want to thank them as well. But I'll wrap it up there and open up to any questions. And hopefully I wasn't too long and explained really where we're going with the iPSCs, but they're a powerful tool. So thank you.
Thanks, Giles. Great talk.
Really
enjoyed it, and I learned a lot too, which is always good. I'll start off with a question. So, what was the shield designed specifically to limit the proliferation of the progenitor stem cells, or was it just something that you discovered when you were using it?
It was a completely discovered thing. The shield itself it's had reiterations of naming. So shield was the most new one that we used. But no, they were purely designed to be injectable and using this cross-linking technology and being able to put the cells in without using these fibronectins or matrigels or vitronectin and all the other compounds and make it safer. And these can be, like I said, made by bacteria and be tailor-made to the patient. And so that was what the proliferation side of it was, a completely unknown thing until we actually did the data, and it suppressed the proliferation. So it's an intriguing result. I'm interested to know if you do suppress the proliferation from the progenitors early, is that detrimental to the graft? Now, if you look to the extent of the growth, it's nowhere near as long. It survives, but it's the growth itself of the neurons that within the cord isn't as long. So do the progenitors play a role in that? I don't know. It's something that I think is fascinating.
Right. So everybody else out there, if you want to ask a question, you can raise your hand. You can ask live, I believe, or you can type any questions you have in the chat or Q&A, and we can relay those questions for Giles. I'll ask another question while I have the mic, so to speak. So when we think about, all the inflammation that happens after brain injury or spinal cord injury, and you put, these type of cells, stem cells in there, does that inflammatory environment, does that influence the way those cells perform or the way they, could the inflammation push those progenitors in a certain direction that's maladaptive?
It could be. I think one of the reasons why I wanted to go down a differentiated route was that if you put neural stem cells in, and this has historically been the case, nearly all of them turn into astrocytes. Nearly about 80-90% will turn into astrocytes. So virtually about, I think it's like 2% will turn into neurons. So the key is like you mentioned, is the inflammatory response has a major effect on what those cells will do. What we know is that the cells that we have, we know the proteomic secretion aspect, and particularly like some of the major hits that we got were MMPs, MMP9, I think it was, and also VEGF was a key aspect to this that came out of all those modularly kind of molecules that will affect the inflammatory response. It's something I'm intrigued to look into, because I think, if Carla Schatz at Stanford was looking into a lot of expression on human cells, whether the progenitors or adult neurons, and MHC class 2 expression and MHC class 1 expression and things. And it's clearly some interest there, is that, and I'd like to look into, is there a benefit of putting differentiated cells in there so it doesn't cause such an inflammatory response that, say, neural stem cells do. It's really untapped. It's really untapped.
And if you put the differentiated cells in there, is that will that have will work in the same way as what's the downside of the differentiated if you put those in?
I don't think there is a downside.
Okay.
I think the stochastic aspect of having neural stem cells and undifferentiated cells is a real problem, because you have no way of knowing what they're turning into. You have no control over it. When you look at the graphs that have been put in neural stem cells in a cord, you have no idea what the phenotype of the neuron is, because most of the labels that they use are UN, just so that's just generally. You don't know if it's glutamatergic, you don't know if it's GABAergic, you don't have any of that information. So my argument was, can we use something that we know more of, and phenotypically we know what it does, and would that give us a better output, and give us a better idea of what it does? It doesn't mean it's right, and people will argue hell for leather is that, are you using the right cell type? Well, in our eyes, the results kind of tell it. It's pretty successful, but also other groups do too, but they don't know what the projection is, and what those neurons are. So I don't have the answer currently, but hopefully we'll get some.
I remember I was at some like a specialty conference in neurology, and someone was showing some spinal cord injury and stem cell work, and I went up to him after his talk and said, "That's amazing." And he was like, "Yeah, well, I didn't show you the tumors."
Yeah. That's a major issue. I mean, it's become it was a huge public argument that two groups had over this problem. They'd done the same. I don't want to name the names, but it became a bit of an issue, is that they had not shown the tumor growth and peel tumors and it forming tumors in the brain, and this other group basically showed it, and it is a problem. And why I'm nervous about using undifferentiated or even partially differentiated cells, that you have that safety issue. And one of the things we're trying to do, there's a couple of ways we're looking at it. We're using two different lines. One's called failsafe, and one is a PAC6-SOX2 lethal knockout iPSC line. So you can basically take out the progenitors at any time you like, whether it's in vivo or before you put them in. And failsafe is the same. It has a kill switch. It's been used in Parkinson's models for iPSC neurons by a group at Melbourne in Australia, and they've successfully done transplants by using this fail-safe line. That they were able to get rid of all the progenitors. So therefore, in their eyes, it's safe for long term because you've got rid of those problems. And I think that is the way to go.
Yeah, makes sense.
Yeah.
All right. Sorry to dominate all the questions. See if there's last chance, I guess, for everybody to either raise your hand or print something in the chat for us.
Either I've confused everybody, or it's
no, I don't think so. You gave a really nice overview. Oh, we have a question from Harry Fu.
Yes, can you hear me?
Yes. Hello, mate.
Okay. Yeah, great talk, very interesting. So one particular, the image you show me after the transplantation of the cell differentiated neurons, and then the IBA1 positive, the microglia looks like significant down, right?
Yeah.
So that's what's your like a potential? Do you want to plan to do something to characterize the microglia, the state change, maybe the neuron microglia crosstalk, and then is it beneficial for this axon regeneration?
Absolutely. It's just not my, I've talked to Jonathan about it. It's not my field. I mean, I get into cold sweats when I talk about information. But I love that field. It's just something that isn't easily answered. So I would love to be able to do that. Yeah. I mean, I think there's immense potential in the crosstalk between neurons and glia, and particularly microglia, in terms of how does the microglia have a major role in turning these guys into regenerating that long? It could be that, the crosstalk between them, whether it's VEGF or other molecules that crosstalk between it. I think there's a huge potential there. It's something that I really didn't explore when I was at Stanford, but I'd love to here because there's so many people who do that work so well. It would be a great thing to do.
Yeah, that would be interesting, especially maybe the synaptic pruning. And
Yeah,
then
absolutely.
Yeah.
Mm-hmm. I think that's something that with the pseudorabies virus marker, I use a rainbow system where if you pre-engineer the neurons, you can use a rainbow system that allows you to see multicolor synaptic contacts between the neurons of interest, which we use. But whether the microglia then prune those connections is a fascinating topic long term in terms of its innovation into those motor neurons caudally. I don't know the answer to that, but I think it would be great.
Yeah, that's good. Thank you.
Thank you, Harry.
Thanks, Harry. That's a good suggestion. I was thinking you could, Harry and I both like some of the single nuclei type approaches. Wonder if you'd be able to identify. I'm sure you would. You'd be able to identify the cells that you put in versus the cells that are there.
Yeah.
But also, I mean, at least from a transcription profile, you'd be able to tell what microglia are doing. I think pretty.
Well, that would be great to do. I think
if you have it in the context of what the neurons and astrocytes and stem cells are all doing together.
And whether that changes over time too, we don't know if that changes from, day 7 to day 14 to day 30 or day 60, we don't know. And particularly just to say is that in terms of proliferation of the progenitors and stuff within the grafts, that due to the way you produce those neurons, you can't currently get rid of, unless you use failsafe or these particular, which I haven't developed totally yet, you can't get rid of these progenitors. And so what's the role of those progenitors in those grafts? We have no idea. Do they interact with the microglia more than the neurons do? We don't know, but it's certainly exciting to think you could do that.
Okay. I'm not seeing it. Does anyone else have any last questions? I'll give you one more chance here. I'm not seeing anything in the Q&A or chat. Kedar, am I missing something?
No, I think that's all the questions we have.
Okay. Well, I guess I'll let everybody get back to their early morning here and have a nice weekend, and look forward to seeing people again at our next NT-RIPS or Buckeye Neurotrauma. Giles, thanks again. Very informative.
Thank you, Jonathan. Thank you, Kedar, and thank everybody else for listening. So
take care.
All right. Take care. Bye, guys.